Hey everyone, welcome back to Brighter Minds Academy's YouTube channel, and in today's lesson we're starting a new unit on genetics. This unit is part of your grade 11 biology course, and it focuses on understanding how traits are passed from parents to offspring. In this genetics unit, we will be learning about topics such as DNA, gene, alleles, chromosomes, meiosis, Punnett squares, Mendelian inheritance, genetic variation, and how genetic information is passed from one generation to the next. However, in today's video, we're going to focus on the foundation of genetics, which is Mendelian's genetic, and we will also introduce one type of Punnett square called the Monohybrid Punnett square. Mendelian genetics is named after Gregor Mendel, who is known as the father of genetics. Mendel was a scientist and a monk who studied inheritance in the 1800s. He wanted to understand how traits are passed from parents to offspring and why offspring sometimes look similar to their parents but not exactly the same. To study this, he used pea plants. Pea plants were a very good choice because they were easy to grow, reproduced quickly, and had traits that were easy to observe, such as tall or short height, purple or white flowers, round or wrinkled seed, and yellow or green seeds. Pea plants were also useful because they can self-pollinate, but Mendel could also control their reproduction by cross-pollinating them himself. This allowed him to decide which plant would be the parent in each experiment. Mendel began with true breeding plants. True breeding means that when the plant reproduces, it always produces offspring with the same trait. For example, a true breeding tall plant always produces tall offspring, and a true breeding short plant always produces short offspring. Mendel crossed a true breeding tall plant with a true breeding short plant to see what would happen in the next generation. The offspring from the first cross are called the F1 generation. When Mendel did this, all of the F1 offsprings were tall. The short traits seemed to disappear completely. Then he allowed the F1 plants to reproduce with each other. This produced the F2 generation. In the F2 generation, the short trait reappeared. About 3/4 of the plant were tall and about 1/4 were short. This observation helped Mendel realize that traits do not blend together. Instead, they are inherited in a predictable pattern. From this experiment, Mendel concluded that some traits are dominant and some are recessive. A dominant trait is the trait that appears when at least one dominant allele is present, whereas a recessive trait only appears when two recessive alleles are present. In Mendel's pea plant experiment, tall was the dominant trait and short was the recessive trait. To explain this using genetics, we use the word gene, which is a section of DNA that controls a trait. Different versions of the same gene are called alleles. For plant height, one allele can represent tall and another allele can represent short. We usually use letter to represent alleles. A capital letter represents the dominant allele and a lowercase letter represents the recessive allele. So for plant height, we use an uppercase T for tall and a lowercase t for short. There are three important combinations of alleles. The two uppercases T means the organism has two dominant alleles, which is called homozygous dominant. If the organism has two lowercases t, means that the organism has two recessive alleles, which is called homozygous recessive. One uppercase T and one lowercase t means the organism has one dominant and one recessive allele, which is called heterozygous. Another important term is genotype, which means the genetic combinations such as two uppercases T, one uppercase, and one lowercase t, or two lowercases t. If we talk about the phenotypes, phenotypes generally mean the physical trait we can actually see, such as a tall or short plant. So a pea plant with the genotype 2 uppercase T is tall. A plant with 1 uppercase T and lowercase t is also tall because the dominant allele masks the recessive one, and only a plant with 2 lowercase t is short. Now, this is where we connect Mendel's experiment to monohybrid Punnett squares. A monohybrid cross is a genetic cross that looks at one trait only. In this case, the one trait is the plant height. So, Mendel's experiment with the tall and short pea plants is an example of a monohybrid cross. A Punnett square is a tool we use to predict the possible genotypes and phenotypes of the offspring. It helps organize the alleles that each parent can pass on. Since Mendel's experiment focused on only one trait, which is the height, we can use a monohybrid Punnett square to show exactly what happened. Let's look at Mendel's first cross, a true breeding tall plant crossed with a true breeding short plant. The genotype of the tall parent is 2 uppercase t, and the genotype for the short parent is 2 lowercase t. We write the tall parent's allele across the top of the Punnett square and the short parent's allele down the side. Then we combine them in each box. Since the tall parent can only pass on 1 uppercase t allele and the short parent can only pass 1 lowercase t allele, every box ends up with 1 uppercase and 1 lowercase allele. This means all of the F1 offsprings are heterozygous, which means they have 1 uppercase t and 1 lowercase t, and because tall is dominant, they are all tall in phenotypes. This matches exactly what Mendel observed in his experiment. Next, let's look at what happened when the F1 generation reproduced. Since all of the F1 plants were 1 uppercase t and 1 lowercase t, Mendel's second cross was between both the heterozygous. This is also a monohybrid cross because we're still looking at only one trait, which is the plant height. To draw the Punnett square, we place one parent's allele on the top, which is 1 uppercase t and a lowercase t across the top, and the other parent's allele, which is 1 uppercase t and a lowercase t down the side. Then we fill in the boxes. So the possible combinations are 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. This gives us a genotype ratio of 1 lowercase t, 1 uppercase t pair, and 2 heterozygous. When we look at the phenotypes, we have 3 tall plants and 1 short plant. So the phenotype ratio is 3:1. This means there is a 75% chance of tall offspring and a 25% chance of a short offspring. This is exactly why Mendel saw about 3/4 tall plants and 1/4 short plant in the F2 generation. This is why Mendel's experiment is such an important example of a monohybrid Punnett square, as it shows one trait can be tracked from parents to offspring using a dominant and recessive allele. It also shows how recessive traits can seem to disappear in one generation and then reappear in the next. Punnett squares help us understand and predict these patterns, which is why they're such an important tool in genetics. Mendel's work became the foundation of modern genetics because it showed that traits are inherited in specific pattern rather than blending randomly. Before Mendel, many people believed that traits mix together, almost like paint, but Mendel's pea plant experiment showed that inheritance is much more organized. His work helped scientists understand the basic rules of heredity and opened the door to modern studies of DNA, gene, and inheritance. To summarize, today we started our grade 11 biology genetics unit by learning about Mendelian genetics. We learned that Gregor Mendel studied pea plants to understand how traits are inherited. He crossed true breeding tall and short plants and observed the F1 and the F2 generation, and discovered the idea of dominant and recessive traits. So this was all in today's video. I hope you enjoyed. If you need extra help for any subject, please contact us on our email address given below. Thank you very much for watching today's video, and I'll see you in the next one. Till then, stay healthy and eat healthy.